So guys, we were discussing human digestive system and we were discussing alimentary canal and its various parts. So we discussed all about small intestine, large intestine. Now this alimentary canal, it has got some glands associated with it. So this alimentary canal plus associated glands, they make the human digestive system. So first we will see the glands which are present in our mouth. Associated glands of alimentary canal which are present in our mouth. As we discussed in the previous lecture, that mouth has got salivary glands in it. In mouth, we have got salivary glands. In humans, there are three pairs of salivary glands. We have got three pairs of salivary glands. These three pairs of salivary glands, they include number first, parotid salivary glands. Then we have got sublingual salivary glands, and we have got submaxillary, or they are also known as submandibular salivary glands. So in humans, there are three pairs of salivary glands present in the mouth. These three pairs of salivary glands, they include parotid salivary glands, sublingual salivary glands, and submaxillary salivary glands. These parotid salivary glands, they are present just below the ear. In humans, they are present just below the ear. Suppose this is the ear. They are present somewhere just below the ear, over here. So these are the parotid salivary glands. These parotid salivary glands, they are a pair of salivary glands which are present just below the ear, right? These glands, parotid glands, they open into the mouth through their ducts known as Stenson's ducts. These Stenson's ducts, they open into mouth opposite the second upper molar in vestibular cavity. These Stenson's ducts, they open into the vestibule of our mouth on the side of second upper molar teeth, right? Then we have got sublingual salivary glands. They are present just below the tongue, at the base of the tongue. So this is a sublingual. It is a sublingual salivary gland. This sublingual salivary gland, it opens on the base of the tongue through its duct known as duct of Ravenous. Ductus of Ravenous. So they open at the base of tongue through ducts of Ravenous. Then we have got a submandibular or submaxillary salivary gland. This submandibular or submaxillary salivary gland, it also opens at the base of the tongue. This is the submaxillary or submandibular salivary gland. It also opens at the base of the tongue through its duct known as Wharton's duct. Wharton's duct. Right? So there are three pairs of salivary glands in our mouth. The pair of salivary glands, which is lying just below the ears on the two sides of our face, they are known as parotid salivary glands. They open into our mouth through their ducts known as Stenson's ducts. These Stenson's ducts, they open into vestibule on the sides of second upper molar teeth, right? Then we have got sublingual salivary glands. As the name indicates, lingua pertains to tongue. So they are present at the base of the tongue. That is why they are known as sublingual salivary glands. They open into the buccal cavity through their ducts known as ducts of Ravenous at the base of the tongue. Then we have got submaxillary or submandibular salivary glands. They are lying just below the mandible. They are lying just below the mandible, the lower jaw bone. They open into the oral cavity through their ducts known as Wharton's duct. They also open at the base of the tongue. So these salivary glands, they produce saliva. They produce saliva. And they produce 1 to 1.5 liters of saliva per day. They produce 1 to 1.5 liters saliva per day. So what is the function of this saliva? It is involved in lubrication of food. Then it has got a salivary amylase in it. That is also known as ptyalin. Salivary amylase. This salivary amylase is involved in the digestion of starch or carbohydrate. We will discuss that in the physiology of digestion, right? So these salivary glands, they produce saliva 1 to 1.5 liters per day, and this saliva contains an enzyme known as ptyalin or salivary amylase, which is involved in digestion of carbohydrates, right? And that will be discussed in the physiology of digestion. So that is all about the salivary glands which are present in our mouth. In pharynx, there are no digestive glands. In esophagus, there are no digestive glands. Then we have got digestive glands in our stomach. Those digestive glands which secrete gastric juice in our stomach, they are known as gastric glands. We will have a look at those gastric glands. The glands which produce gastric juice in our stomach, they are known as gastric glands. We will have a look at those gastric glands. As I told you earlier, in our stomach there are some approximately 35 million gastric glands. And these gastric glands, they produce the gastric. Most of the gastric juice is produced by gastric glands which are present in the fundus. So they are known as fundic glands. Those fundic glands, they are mostly involved in secretion of the gastric juice containing enzymes. So each gastric gland is a tubulo-alveolar gland. Each gastric gland is a tubulo-alveolar gland. That means in part it is tube-like and in part it is sac-like, right? So I will draw here the tubulo-alveolar. So in part it is tube-like and in part it is sac-like. That is why they are known as tubulo-alveolar. So this is the tube-like part and these are the sac-like parts. So these vase-like cells, they are the mucus-producing cells. These vase-like cells, they are mucus-producing cells. And in between them, there are other cell types. So these vase-like cells, they are known as goblet cells. They are goblet cells. These somewhat mushroom-shaped cells, raised mushroom-shaped cells. They are known as parietal cells. They are known as oxyntic cells. Oxyntic cells, or we also call them as parietal cells. Parietal cells. Then these numerous cells, which are most abundant, they are known as chief cells, or we call them as peptic cells, or sometimes they are known as zymogen cells. Then at the base of these gastric glands, there are certain endocrine cells, enteroendocrine cells. These enteroendocrine cells, they are the Paneth cells. So this is a particular gastric gland. This is a particular fundic gland. This is a fundic gland. So this fundic gland, it produces the gastric juice and it also produces the mucus. So this is the mucus. This mucus, it lines the lumen of stomach internally. This mucus protects the stomach from the action of HCl and action of protein digesting enzymes. In gastric juice, HCl is released. In gastric juice, protein digesting enzymes are released. Since the wall of stomach is muscular, so these protein digesting enzymes, they could have easily digested the muscular wall of stomach. HCl could have easily eroded. HCl could have easily damaged the wall of stomach. But what protects the stomach from the action of its HCl, from the action of its own protein digesting enzymes? So it is this mucus which is produced by these goblet cells, right? So these goblet cells, they produce mucus. This mucus lines the stomach internally. It lines the luminal surface of stomach internally, and it protects the stomach from the action of HCl and action of digestive enzymes, as this mucus is highly resistant to the action of HCl and protein digesting enzyme. Then in this gastric gland, we find oxyntic cells. Oxyntic cells, they release HCl. So mucus is produced by number one cell, goblet cell. Then number two cells, oxyntic cells or parietal cells. Oxyntic cells or parietal cells. They produce HCl. This hydrogen chloride, it is a corrosive acid. It could have easily damaged the wall of stomach, but its action is protected by stomach is protected from its action by mucus, right? So oxyntic cells or parietal cells, they produce HCl. Then we have got the third type of cell, known as chief cells or peptic cells or zymogen cells. They produce third type of cells, chief cells, peptic cells or zymogen cells. They produce the proenzymes. Proenzymes in gastric juice, there are two proenzymes: pepsinogen and prorenin. And prorenin. Pepsinogen and prorenin. So these two proenzymes, they are released by chief cells, peptic cells, or zymogen cells, and they also produce and they also release a weak gastric lipase. They also release a weak gastric lipase, right? They also release a weak gastric lipase. Then we have got fourth type of cell, Paneth cell. These Paneth cells, they release lysozyme into gastric juice. This lysozyme, as you know, it is an antibiotic. It is an antibiotic, natural antibiotic. So it kills the pathogens, it kills the bacteria which may have entered along with ingested food, right? So this is the structure of a gastric gland, and its main four cell types are goblet cells, oxytic cells or parietal cells, chief cells, chief cells, peptic cells or zymogen cells, and Paneth cells. So goblet cells, they produce mucus. We discussed the role of mucus. Oxyntic cells or parietal cells, they release HCl. This HCl, it is involved in activation of these proenzymes, pepsinogen and prorin. It carries out, HCl carries out autocatalysis of pepsinogen and prorin. HCl converts pepsinogen into pepsin and prorenin into renin, right? So it is involved in autocatalysis of these proenzymes. And it provides strong acidic medium in the stomach for the digestion of protein. Pepsin and prorenin. Pepsin and renin, sorry. Pepsin and renin, they work in strong acidic medium. So HCl provides a pH of 1.2 to 1.8. It generates a pH of 1.2 to 1.8 in stomach, which is suitable for the action of protein digesting enzyme. Then we have got these chief cells, peptic cells or zymogen cells. They release these proenzymes, pepsinogen and prorin, which are involved in protein digestion. And it also releases a weak gastric lipase. So these chief cells, peptic cells or zymogen cells, they also release a gastric lipase, which carries out some little digestion of fat in the stomach. The amount of digestion of fat in stomach is very negligible. Negligible amounts of fat are also digested in stomach by this gastric lipase, but it is not strong enough. Lipases work best when the fats are first emulsified. Since in stomach the fats are not emulsified, emulsification of fats that occurs in small intestine, right, by the help of bile and pancreatic juice. Since emulsified fats are not present in stomach, so this gastric lipase, it is not able to carry out digestion to a significant level in stomach, right? So in stomach, very little quantities of fat may get digested. Then we have got Paneth cells at the base of these glands. These Paneth cells, they produce a natural antibiotic lysozyme in the gastric juice. So this is a fundic gland. This is a fundic gastric gland. Most of these digestive juice, most of these gastric juice producing glands, they are located in fundus. So we call it a fundic. So we say it is a fundic gastric gland. Gastric gland. So this is a fundic gastric gland. So that is all about the composition of gastric gland in the stomach. Now after this, after stomach starts the element in small intestine. After stomach starts the small intestine. You know, in small intestine, the first juice which is secreted in its lumen is bile and pancreatic juice. Small intestine in its duodenum receives the digestive juices from liver and pancreas. We have discussed where the liver is, how the liver releases bile into duodenum and through which duct. So I have told you our liver, human liver, it is four-lobed, but there are only two large lobes which are significant in secretion of this bile. They are known as right lobe, right hepatic lobe, which is the largest lobe, and then there is a left hepatic lobe, which is the smaller lobe. Two small lobes, caudate lobe and quadrate lobe. They are lying just below the right lobe. So from the lobes of liver, hepatic duct arise. Hepatic ducts join together, they form common hepatic duct. Then common hepatic duct joins with the duct of gallbladder, known as cystic duct. So when cystic duct and common hepatic duct join together, they form common bile duct. Then that common bile duct combines with the duct of pancreas to form hepatopancreatic duct. Then hepatopancreatic duct opens into duodenum through the ampulla of Vater, which is guarded by sphincter of body, right? So liver produces bile and pancreas produces pancreatic juice. What are their functions in the digestion? We will discuss that in physiology of digestion, right? So in the duodenum, bile and pancreatic juice is released by liver and pancreas. Then we go into other parts of small intestine. In the wall of small intestine, there are certain glands known as intestinal glands. Intestinal glands. These intestinal glands, they are also known as crypts of labor. Crypts of labor. They release these crypts of labor. They release intestinal juice. They release intestinal juice known as succus entericus. Succus entericus. They release intestinal juice known as succus entericus. This succus entericus, it contains carbohydrate digesting enzymes, it contains protein digesting enzymes, and it also contains some lipid digesting enzymes, fat digesting enzymes, right? So we will see these crypts of labor when we will draw the diagram of the wall of alimentary canal. Now we are going, after this, I am going to discuss the histology of alimentary canal. So I am making a diagram of the wall of alimentary canal. In that diagram, I will show you where the crypts of labor are located. And these crypts of labor they release intestinal juice, or also known as succus entericus. So first we will discuss the histology of alimentary canal. We will see the structure of the wall of alimentary canal. Now we will discuss histology of alimentary canal. So if we cut a section of alimentary canal through the intestine, at the level of intestine, what we will see? What do we see there? This alimentary canal, it is externally covered by a layer of connective tissue and epithelium. This wall of intestine, wall of alimentary canal, it is externally covered by a layer of connective tissue fibers, and epithelium. This epithelium is derived from mesoderm. So this epithelium is known as mesothelium. This epithelium is known as mesothelium. And beneath this mesothelium, there are certain connective tissue fibers. So they are together known as, we call this first layer of alimentary canal, we call it as tunica serosa. Tunica serosa. Or it is known as serous membrane. It is also known as serous membrane. Serous membrane. So our alimentary canal is externally covered by a layer of epithelium and connective tissue fibers. So that layer of epithelium is derived from mesoderm. So we call it as mesothelium. Epithelium which is derived from mesoderm, that is known as mesothelium. And it is having some connective tissue fibers also associated with it. So these connective tissue fibers and this mesothelium, it is together known as tunica serosa or serous membrane. So they form the outermost covering of the alimentary canal. This tunica serosa, it is followed by a layer of muscles. So this layer of muscles, it is known as tunica muscularis. Tunica muscularis. So first was tunica serosa, second is tunica muscularis. This tunica muscularis, it is formed of two types of smooth muscles. These smooth muscles, they run along the length of the alimentary canal. So they run along the length of alimentary canal. So these smooth muscles which run along the length of alimentary canal, they are known as longitudinal muscles. So these are the longitudinal muscles. And these muscles, which are circular, they are present along the circumference of alimentary canal. These muscles are present along the circumference of alimentary canal. They are known as circular muscles. Circular muscles. So tunica muscularis, it is formed of longitudinal muscles and circular muscles. So this layer of longitudinal muscles and circular muscles, which is present in the wall of alimentary canal, it is known as tunica muscularis. And you can see in between longitudinal muscles and circular muscles, there is a network of nerve fibers, some neurons and nerve fibers. This network of neurons and nerve fibers, which is present between longitudinal muscles and circular muscles, it is known as plexus of. It is known as plexus of Orbit. It is known as plexus of Orbit, or it's also known as myoenteric plexus. We also call it as myoenteric plexus. So between longitudinal muscles and circular muscles, there is a network of some neurons and nerve fibers. This network of neurons and nerve fibers, which is lying between longitudinal muscles and circular muscles, it is known as plexus of Orbit, or it is known as myoenteric plexus. It controls the movement of muscles of the alimentary canal. You know, alimentary canal shows peristaltic movements, it shows segmental movements, right? So all the muscle contraction which occur in the wall of alimentary canal, they are controlled by this plexus of Orbit, and this is another plexus known as plexus of Messenger, right? So then between below the circular muscle, this is the layer of circular muscle. Below this layer of circular muscle, there is another network of nerve fibers and neurons. This is known as plexus of. This is known as plexus of Messenger. This is known as plexus of Messenger. So just below tunica serosa, we have got tunica muscularis, formed of two types of muscles: outer longitudinal muscle layer and inner circular muscle layer. Between longitudinal muscle layer and circular muscle layer, there is a network of nerve fibers and some neurons. This network of nerve fibers and neurons, which is known as plexus of Orbit or myoenteric plexus. Then just below the layer of circular muscles, we have got another network of neurons and nerve fibers. That is known as plexus of Messenger. So this plexus of Orbit and plexus of Messenger, they together control the muscular movements of alimentary canal. So peristalsis, segmental movements, right? They are all controlled by this network of nerve fibers and neurons, right? Then below this tunica muscularis, there is a layer of loose connective tissue. This layer of loose connective tissue, which has got various blood vessels, lymphatic vessels in it. This layer of loose connective tissue, which has got most of the blood vessels and lymphatic vessels in it. This is known as tunica submucosa. So this layer of connective tissue. It is known as, it is a third layer of wall of alimentary canal. We call it as tunica submucosa. Submucosa. So this tunica submucosa, it contains blood vessels and lymphatic vessels and loose connective tissue fibers in it, right? Then below this, below tunica submucosa, there is a thin layer of longitudinal muscle fibers again. Then inner to longitudinal muscle fibers, there is a thin layer of circular muscle. Then inner to them, there is connective tissue. So this layer from here to here, it is known as tunica, which is the fourth layer. It is known as tunica mucosa. Together known as tunica mucosa. So this is tunica mucosa. And in this tunica mucosa, there is a layer of longitudinal muscles and circular muscles. This thin layer of longitudinal muscles and circular muscles, which is present in tunica mucosa, it is known as muscularis mucosa. Muscularis mucosa. Inner to muscularis mucosa, there is a layer of connective tissue. This layer of connective tissue, which is present inner to muscularis mucosa, it is known as lamina propria. It is lamina propria. And this lamina propria, along with its lining epithelium, it is drawn into finger-like outgrowths. These finger-like outgrowths, they are the villi. They are the villi. And these villi are lined by brush-bordered columnar epithelium. They are lined by brush-bordered columnar epithelium. Right? So in this way, we find that wall of alimentary canal, it is formed of four distinct layers. Wall of alimentary canal is formed of four distinct layers of tissues. The outermost wall of alimentary canal, if this is alimentary canal is a tube-like structure. Suppose this is our alimentary canal. It is lying inside our abdominal cavity, right? So this outer surface of alimentary canal, it is covered by a layer of mesothelium and connective tissue fibers. this layer of mesothelium and connective tissue fibers. This is known as tunica serosa, or we call it as serous membrane. So outermost covering of wall of alimentary canal is formed of mesothelium and connective tissue fibers known as tunica serosa or serous membrane. Inside this tunica serosa, there is a layer of longitudinal muscles and circular muscles. So this layer of longitudinal muscles and circular muscles, it forms tunica muscularis. It forms tunica muscularis. In this tunica muscularis, there is outer longitudinal muscle layer and inner circular muscle layer. In between longitudinal muscle layer and circular muscle layer, there is a network of some nerve fibers and some neurons. This network of nerve fibers and neurons, which is present between longitudinal muscles and circular muscles, we call it as Plexus of Orbit. This network of nerve fibers, it is known as Plexus of Orbit, or we also call it as myoenteric plexus. Then just below the circular muscle layer and tunica submucosa, between tunica submucosa and circular muscle layer, there is another layer of nerve fibers and some neurons, right? So this another network of nerve fibers and neurons, this is known as plexus of Masonry. So there are two plexuses present. One is known as plexus of Orbit. It is present in the tunica muscularis between longitudinal muscles and circular muscles. Another is known as plexus of Masonry, which is lying between tunica muscularis and tunica submucosa. It is lying between tunica muscularis and tunica submucosa. So this is the circular muscle layer. In between this circular muscle layer and tunica submucosa, there is another network of nerve fibers. This is known as plexus of Masonry. So both this plexus of Orbit and plexus of Masonry, they control the muscle movements of the alimentary canal, and they also control some sensory functions of the alimentary canal. In addition to controlling the muscle movement of alimentary canal, they also control some sensory functions of the alimentary canal. So they regulate the muscle movement, the movements of intestines, movements of alimentary canal, and they also regulate sensory functions. Then just below tunica muscularis, we have got a layer of loose connective tissue fiber. This layer of loose connective tissue fiber, which is having blood vessels and lymph vessels in it, right? Or nerve fibers in it. This is known as tunica submucosa. So outermost layer is tunica serosa, then there is tunica muscularis, then there is tunica submucosa. Inner to tunica submucosa, we have got tunica mucosa. Inner to tunica submucosa, we have got tunica mucosa. So from here to here, this is all tunica mucosa. We can see this tunica mucosa. We can divide it into further three layers. Muscularis mucosa, lamina propria, and the lining epithelium. So tunica mucosa, it can be divided into three more layers. The outer layer of longitudinal muscles and circular muscles. They are very thin. In the tunica muscularis, this layer of muscles is very thick. But in this tunica mucosa, this outer longitudinal muscle layer and inner circular muscles, they are in the form of a very thin layer. So this is known as muscularis mucosa. Then inner to muscularis mucosa, there is a layer of loose connective tissue that is known as lamina propria. And then this lamina propria, it is surrounded by epithelium and is drawn into finger-like projection. So these finger-like projections, which are surrounded by brush-bordered columnar epithelium, they are known as villi. In these villi, we find there are small lymph vessels known as lacteals. In these villi, there are small lymph vessels known as lacteals. These are the lacteals in villi. Lacteal. And these lacteals are surrounded by blood capillaries also. So in villi, there are a large number of blood capillaries also. So they help in the absorption of food. So digested nutrients, they are present in the lumen of alimentary canal. So from the lumen of alimentary canal, those digested nutrients, they enter into blood. So they are first taken by brush border columnar epithelium. The nutrients are taken by brush border columnar epithelium. And this brush border columnar epithelium releases those nutrients then into lymphatic circulation or into capillaries of the villi, into the capillaries of villi, right? So that is all about the histology of alimentary canal. In stomach, tunica muscularis is formed of another layer of muscles. In stomach, there is another layer of muscles. In addition to longitudinal muscles, circular muscles, there is another layer of muscles that is known as oblique muscles. In stomach, we find tunica muscularis is formed of three layers. In stomach, we find this tunica muscularis is formed of three layers. In stomach, tunica muscularis, it is formed of three layers: outer longitudinal muscles, middle circular muscles, and innermost oblique muscle. Oblique muscle. So in the wall of stomach, this tunica muscularis, it is formed of three layers instead of two layers: longitudinal muscles, circular muscles, and oblique muscles. Right? Then, if we look at in the wall of colon, longitudinal muscles, they are accumulated in the form of three longitudinal bands. In colon, these longitudinal muscles of tunica muscularis, they are aggregated in the form of three longitudinal bands. In colon, longitudinal muscles of tunica muscularis, they have aggregated in the form of three longitudinal strips. These three longitudinal strips of muscle fibers, they are known as tinea coli. They are known as tinea coli. So in colon, these longitudinal muscles of the wall of alimentary canal, they have aggregated in three longitudinal bundles of fibers. These three longitudinal bundles of fibers, which run in the wall of colon, they are known as tinea coli. And they produce bulges on the wall of colon. They produce bulges on the wall of colon. These bulges, these pocket-like outgrowths, which are developed on the wall of colon, they are known as hostra. Hostra is a plural word. Hostrum is singular. Right? So they give rise to hostra in the wall of colon. Right? So in the remaining part of alimentary canal, longitudinal muscles, they are present along the circumference. In the remaining part of wall of alimentary canal, longitudinal muscles, they are present along the circumference. Right? But in colon, these longitudinal muscles are not present along the circumference. They have aggregated, they are accumulated into three longitudinal bundles. This is one longitudinal bundle of muscle fibers. This is another, this is another. So these longitudinal bundles of muscle fibers, which are present in the wall of colon, they are known as tinea coli. They are known as tinea coli. And the remaining part of this wall of alimentary canal, it projects out in the form of pocket-like outgrowths. So these pocket-like outgrowths, which are present in the wall of colon, they are known as hostra. Singular is hostra, and their plural is hostra. So these hostra are the bulgings of the wall of colon. So when I draw this diagram of colon, if you remember, I had shown there pocket-like outgrowths in that colon. So when I draw the colon, I drew the colon like this. I draw the colon like this. I have shown colon like this. So what are these pocket-like outgrowths? These pocket-like outgrowths on the wall of colon, they are the hostra. These are the hostra. These are the hostra. So they increase the surface area of this colon. Colon is a very short structure. It is just 1.5 meter in length. So it has to absorb, it has to reabsorb water and some minerals and some vitamins from undigested food material. So its wall is thrown into these pocket-like outgrowths. It enhances the absorptive surface of the colon so that this water, minerals, and vitamins they are efficiently reabsorbed from the colon, right? So that is all about the histology of alimentary canal. So I think this is sufficient for today. Thank you.